Hi students, in the present lecture we will be discussing the nerves. What is a nerve, how the nerve is formed, followed by amazing topic called as neuroglia. Clear student? So group of cytons in CNS is called as nucleus. Group of cytons in the peripheral nervous system is called as ganglia. That is, when the group of cytons, soma, we have discussed in the previous session what is a cyton, isn't it? So group of cytons, a bundle of cytons, if it is present in the central nervous system, that is brain and spinal cord, is called as nucleus. And group of cytons in the peripheral nervous system, PNS, that is called as ganglia. Next one is, this is the first point. The second point is the group of axons, that is nerve fibers. Nerve fibers. The group of axons in the central nervous system is called as tracts. The group of axons in the peripheral nervous system are called as nerves. In this way, nerves here. The same group of axons, that is nerve fibers, when present in the central nervous system, that is called as tracts. The group of axons, the peripheral nervous system, that is called as the nerves are formed here. Now these nerves, how the nerve fiber, you see here the nerve fiber. What is a nerve fiber? The nerve fiber means the axon. Axon means the nerve fiber here. Nerve fiber. See here the individual nerve fiber. This is a nerve fiber. The nerve fiber is enclosed by a loose connective tissue sheath, and that's called as endoneurium. So what is an endoneurium? Endoneurium means the loose connective tissue sheath around the nerve fiber, here called as axon, that is called as the endoneurium. Like this, many such things are formed here. For example, here, many such things are formed. Okay, here, many are there here. Here form, here. All these forms the bundle, nerve bundle called as a fascicle. What is this called as a fascicle? Also called as a fasciculus. Fasciculus. So axon means a nerve fiber. The nerve fiber, this is the nerve fiber, enclosed by the connective tissue sheath, loose connective tissue sheath, and that's called as an endoneurium. Many such things are formed here, like this, here many structures are formed. All these groups, the nerve fiber along with the endoneurium, nerve fiber along with the endoneurium, nerve fiber along with the endoneurium, like this, many are formed. All these bundles are there here. This bundle is called as a fascicle, which is also called as a fasciculus, here. So like this here, many fascicles are formed here. All these together in turn forms the another larger structure here called as a nerve. That is called as a nerve. Now the fascicle is enclosed. This is a fascicle now. This is a fascicle, isn't it? The fascicle is enclosed by another connective tissue sheath called as the perineurium. And all these fascicles along with the perineurium many are there, it forms a nerve. The nerve is enclosed by another connective tissue sheath called as a epineurium. Epineurium here. So like this, each nerve fiber enclosed by connective tissue sheath called as endoneurium. Many such structures are there. This forms a fascicle or fasciculus, which is enclosed by connective tissue sheath called as a perineurium. Like this, many structures are formed here. This forms a nerve bundle called as nerve here. The nerve is enclosed by another connective tissue sheath called as a epineurium. In this way, there is a structural arrangement here. Clear, students? So let's discuss now. I told you that nervous tissue nervous tissue. Two types here. One is neurons, conducting cells, and non-conducting cells are called as neuroglia. In the previous session, we had discussed neurons, which are the conducting cells. Now the non-conducting cells, which are not responsible for the conduction of the action potentials, they are called as the neuroglia. So these neuroglia are nothing but non-conducting cells, which are nothing but the supporting cells of the nervous tissue. They are not participating in the conduction of the action potentials. That's why they are called as non-conducting cells, which are nothing but the supporting cells, here. You're following, students, all of you. So let's discuss now the neuroglia present here. These neuroglia are present in the CNS, and the neuroglia are also present in the PNS. So neuroglia of CNS, neuroglia of PNS. Let's discuss central nervous system, neuroglia of the peripheral nervous system. Let's discuss now. Clear, student? But show all of you. You're enjoying now, all of you. First of all, neuroglia of CNS, central nervous system. Clear, but show. What is that? Just remember a code, OMIA. The cells which are present in the neuroglia of CNS comes under OMIA. O stands for oligodendrocytes, and M means here microglia. E for ependyma or ependymal cells, and A for astrocytes here. Let's discuss one by one in detail here. So these are the neuroglia of a central nervous system. First of all, starting with the oligodendrocyte. Cyte, what is an oligodendrocyte? Cyte means a cell. Okay, in the previous session I told you that the myelin sheath is formed because of the oligodendrocyte. A single oligodendrocyte can myelinate many axons. It can secrete the myelin sheath around many axons here. That's a function of the oligodendrocyte. Myelin sheath, which is a lipid structure, is formed by the oligodendrocyte. A single oligodendrocyte can form myelinate many axons. See here, this is a axon. Here the sheath is formed, myelin sheath. This sheath is formed by the oligodendrocyte. All of you see here. So this is a neuron. This one is a neuron, cyton. This is a cyton. This is a nucleus. So this is a neuron. Can you see, all of you? This is a neuron. And here is a myelin sheath formed. So myelin sheath. This myelin sheath is formed by the oligodendrocyte. So this single oligodendrocyte can myelinate many axons. That's a function of the oligodendrocyte. This is finished. Next one is microglia. In the previous lectures, I was discussing what is by mononuclear phagocytic system. That is WBC leukocytes, because of diapedesis, they can go to the extravascular spaces by the process called as diapedesis. And if they go to the different places, when they are present in the nervous tissue, they are called as microglia. When they are present in the nervous tissue, the mononuclear phagocytic system, the WBC, when they are present in the nervous tissue, they are called as microglia. Now the microglia are the only cells of the nervous tissue which are derived from mesoderm. In the rest of the nervous tissue is derived only from ectoderm. Entire nervous tissue, nervous system is derived from ectoderm, whereas microglia are the only cells of the nervous tissue which are derived from mesoderm. It's a very, very important bit. Why? Because it's a part of the connective tissue. Actually, they are for nothing but WBC, isn't it? Blood, mesodermal in origin. And that's why these are also mesoderm. They're nothing but the phagocytic system. Why called as mononuclear phagocytic system? They have formed the mononuclear phagocytic system. Phagocytic system. What is the mononuclear? Single nucleus is there, and they are the they comprise the phagocytic system because they are the phagocytes. What is phagocyte? Which can engulf and kill the bacteria is called as phagocytosis. The cell which can engulf and kill the bacteria is called as a phagocyte. So these are the WBC are the phagocytes here, isn't it? So here these are the phagocytes and comprise a mononuclear phagocytic system. So whenever the nervous tissue is having more injuries and infections, when the nervous tissue is having the bacterial infection, for example, viral infection, for example, then the microglia are activated, the WBC are activated, and they go to the extravascular spaces to the nervous tissue, and they are formed into the cells, special cells called as the microglia. Okay, and they can engulf and kill the bacteria. In this way, the nervous tissue is free of any bacterial infection because of the microglia, and they are mesodermal in origin. They are the only cells of the nervous tissue which are mesodermal in origin here. They form the part of the mononuclear phagocytic system. I have made an earlier video. I'll describe the link in the description box. Next one is ependymal cells. What is by ependymal cells? Let's discuss now. You know this first there are 4 brain ventricles are there. Brain ventricles. Brain ventricles means brain cavities, the empty spaces of the brain. They're called as brain ventricles or cavities. The hollow spaces are there. There are 4 brain ventricles are there. 4 ventricles are there. First 2 are called as present here. Right and left ventricles. Right and left ventricles called as paracells. Right ventricle is present in the right cerebral hemisphere, and left ventricle is present in the left cerebral hemisphere. They are called as lateral ventricles or paracells. And these are called as first ventricle, this is second ventricle, and the third ventricle is present called as the diaceal. Diaceal is present in the diencephalon. And fourth ventricle is called as metacele or myelocele, present in the medulla oblongata, So these are the 4 brain ventricles called as brain cavities. And next one, this is the first point. The second point is this spinal cord. Spinal cord contains a cavity called as the central canal. Central canal is a cavity of the spinal cord. There is a common fluid which is flowing throughout the 4 brain ventricles and the cavity of the spinal cord. There is a common fluid flowing. This common fluid is called as a cerebrospinal fluid. Cerebrospinal fluid. Why is called as cerebro? Cerebro means brain. Spinal means spinal cord. There is a common fluid which is flowing throughout the brain. That is brain ventricles, 4 ventricles, and also it is going through the which one? The spinal neurocele, also called as central canal of the spinal cord. That fluid is called as cerebrospinal fluid or commonly called as CSF. Now, which one is driving these 4 common fluid throughout the brain ventricles and throughout the spinal cord? What is the nature here? That is called as a ependyma. Also called as ependymal epithelium. They are nothing but, ependyma is nothing but ciliated columnar or ciliated cuboidal epithelium. Ciliated columnar or ciliated cuboidal epithelium. Why ciliated? Because it, as the name indicates, it contains a cilia. This is a columnar epithelium or cuboidal epithelium, for example. This is a columnar epithelium, and it contains a free surfaces, contains a cilia. Okay, these are called as a ependyma. Can you see all of you? These are the ependymal epithelium or ependymal cells or ependyma. They are lining the cavities, 4 cavities of the brain. That is brain ventricles, and also cavity of the spinal cord called as a spinal neurocele or central canal. The ependymal is by the help of the cilia. The cilia flushes out here, drives this fluid throughout the brain ventricles and also driving into the spinal cord. That is central canal of this spinal cord here. By the help of this cilia, the fluid is driven here throughout the 4 brain ventricles and also driving into the central canal of the spinal cord here. That's the function of this one here. So if there is a dysfunction, if there is a lack of functionality in the ependymal epithelium, it results in the disorder called as hydrocephalus. Hydrocephalus. Yes, all of you type in the comment box below what is hydrocephalus. Lack of this ependymal function. When the ependymal epithelium becomes dysfunctional, then there is a disease called brain disorder called as hydrocephalus. What is hydrocephalus? All of you type in the comment box below. So that's all about the ependymal myocho. All of you. Next one, let's discuss about the astrocytes. Astrocyte. Astro. Anything astro means star. Because as the name indicates, they are star shaped here. Can you see all of you? These are the astrocytes. So these are the astrocytes. This is a microglial cell. Okay, this is a oligodendrocyte. Astrocytes here drawn in the red color. Star shaped cells present throughout the nervous tissue here. There are supporting cells, very, very important in the nervous tissue. Why? Because they form the blood-brain barrier. Blood-brain barrier. For example, here is a blood representation. Here is a capillary which represents the capillary, for example. And this is a representation of the nervous tissue. Neuron, for example, this is a representation of the neuron. So here is a barrier formed here. Here is a barrier formed. This is the blue color here. This is the barrier formed called as astrocyte. Astrocyte here. That is, this is the capillary, blood capillary. This is the neuron. Neurons, total brain comprises only just 2% of the human body weight. Brain comprises just 2% of the human body weight, but it consumes 20% of oxygen. 20% of oxygen. That is, more metabolism is there because more intense neuronal activity is there, isn't it? That's why it consumes as much as 20% of total oxygen. More metabolism. That is, oxygen should be diffused readily into the nervous tissue. And brain contains as many as 100 billion neurons. 100 billion neurons. That is, intense neuronal activity is there and they should be readily supplying the oxygen. And glucose also here. Because brain takes the glucose and other structures here. Water, glucose, oxygen should be readily supplied to these 100 billion neurons around present in the brain. And which one is doing here? That is done by the astrocyte here. So here is a capillary. The capillary is containing the blood. The blood is containing the oxygen, glucose and water, of course. And they must be readily supplied to the brain to which one? Neuron. Through which one? Astrocyte. Why? You may ask the question, sir, why astrocyte is there? It can be directly supplying here. Because you see here is a crucial point to be remembered. Astrocyte shouldn't be allowing this kachra. Only the essential particles. What are the essential particles here? Oxygen, glucose, water and other essential particles which are readily necessary for metabolism of the neuron here. Only those particles should be selectively supplied. Not the kachra, not the unnecessary and useless particles shouldn't be supplying to the neuron because they should be having the intense neuronal activity must be there. So only the supply of the essential particles should be there into the neuron here. And that's the duty of this astrocyte. That's why it forms a barrier between a hurdle between the capillary, that is a blood, and the brain here, that is called as here neuron. That's why we can say that it is a blood-brain barrier, astrocyte. Many astrocytes, the star-shaped cells are present throughout the nervous tissue. This is the activity here. So these omia, they form the neuroglia of the central nervous system. Next one is neuroglia of the peripheral nervous system. Let's discuss now. Neuroglia of the peripheral nervous system. S and S. That is a code to be remembered here. Double S. Omia for Neuroglia of CNS, Neuroglia of PNS, SS. S for Schwann cell and S for satellite cell. Satellite cell. Schwann cell we already discussed now, all of you. What is a Schwann cell? Here is the axon and here is the myelin sheath present here, isn't it? Myelin sheath, myelin sheath, myelin sheath. And here this is called as nodes of Ranvier. Nodes of Ranvier. And this is the myelin sheath. Myelin sheath. Okay, I am rubbing this nodes of Ranvier here. And outside the myelin sheath, what is the cell outermost structure present? Neurilemma. Neurilemma. Neurilemma and myelin sheath put together called as a Schwann cell. Schwann cell. So what is a Schwann cell? The myelin sheath, the lipid coating sheath, okay, which is present around the axon is called as myelin sheath, and outside the myelin sheath, that is a neurilemma which contains a nucleus and the cytoplasm, that is the outermost layer of the axon here, isn't it? Neurilemma. So myelin sheath along with the neurilemma comprises a Schwann cell. Schwann cell here. You're following, all of you. So that's next one is a satellite cell. The satellite cell is present around the cyton. See here, the cyton. Okay, rough diagram here. OD ribbon here. So example, this is a cyton. Around the cyton here, a small cell is there. That's called as a satellite cell. The satellite cell supports the cyton. Satellite cell supports the cyton and not only supports but also sends the essential particles to the cyton here. Satellite, why is called? Because it's a supporting in nature. That's why it's a satellite cell present around the cyton, and they support, not only support but also supply the essential particles to the cyton. In this way, the satellite cell is supporting the cyton here. These comprises S and S, Schwann cell and satellite cell. They form the neuroglia of the peripheral nervous system. That's all for present day lecture, my dear bacho. Hope you enjoyed my session. Please type in the comment box below. You'll like it if you dearly support this one. All of you, bye, all of you students.